My name is Ignacio Bianco. I'm an intern here at the Intocombit World Organization, and I'm going to be presenting this paper titled Bacillus Calmette-Guerin Treatment of Bladder Cancer, a Systematic Review and Commentary on Recent Publications. It was published by three physicians of UT Health San Antonio, and it was published in the journal Current Opinion Urology. This journal is specialized for urological treatments, and it has a relatively high impact factor and H index within the field, meaning that we can expect a high level of rigor from this publication. Before I move on and discuss what findings and what information they managed to review on this paper, I'd like to just briefly introduce how BCG treatment works from a more specific point of view. First of all, BCG is indicated for treatment of bladder cancer in situ, meaning it hasn't invaded the basement membrane, and prevention of the relapse of some other types of bladder cancer, mainly T1, meaning that it has not invaded any other adjacent structures or the submucosa or anything like that. So very mild early cases of bladder cancer. The way in which it works, and I'm going to show you here very quickly, is that through a catheter, a BCG solution is injected inside of the bladder. This is called instillation. And usually the way treatment works is that there's an induction course of 6 weekly treatments or so, and a maintenance course, which is going to be followed up to 3 years after the induction. And yeah, that's the basis of the BCG treatment. Before I move on with the most important of the paper's findings, I want to just briefly mention something that I found particularly important from the methods section, which is that although in the past years, in the past 15 years or so, there has been a plateau on the publications of BCG, there's still interest within the field of BCG, as we can see here by the number of publications of both BCG specific papers and BCG papers in relation to bladder cancer. What this basically means, what's being underscored here, is that there's still need for further exploration, right? In terms of therapeutic effects, response prediction, and so forth. And this is precisely what this paper aims to address, which is basically the gaps in research and the potential behind BCG treatments. And to start, what the paper addresses are the different kinds of strains that they are, and the research that has been published based on their clinical efficacy. So just to give a brief introduction, all of these strains come from one original strain from Mycobacterium bovis, which was first cultured by Dr. Guerin, which was the physician that named this bacterium. Then afterwards, this strain got disseminated into different labs throughout the world, and some new strains were developed from this original strain, meaning they all share the same basic mutations, but they differ in some of their cell wall components, which is going to dictate their immunogenicity, or the degree to which these strains elicit an immune response, which is one of the main underlying pillars of BCG treatment. Nowadays, for BCG, we're using three main strains. And what this paper shows is that some strains show better potential in terms of treatment efficacy than others. But research around it is still insufficient. For example, they signal that this Connaught strain, which supposedly has the highest immunogenicity, because this immunogenicity is dictated by components in their cell wall and cell envelopes. As I said, this diagram here is taken from a paper published on tuberculosis, but still some of the main components are shared amongst both mycobacteria, mainly LAM, acyl lipids, mycolic acids, and so forth. And the precise composition of these components, as well as some specific moieties, which really transcend the scope of this publication, are the ones that dictate its immunogenicity. The one we're using most often today is TICE, which is supposed to have the lowest immunogenicity. And still, evidence showing that Kona has better so clinical outcomes than TICE is still insufficient and needs to be studied further. Though it is important to point out that apparently due to some manufacturer issues or an issue along those lines, Kona is no longer used in the US. But in some other parts of the world, I think in Japan, the strain Tokyo 172, which is supposed to have a sort of intermediate immunogenicity, is being used. And there are hints that it could possibly yield better outcomes than TICE, though this still, as I said, needs to be further reviewed. Now, next, they wanted to know whether there were some predictive factors related to BCG treatment. And for that, they based themselves on the concept of priming, as they saw that subcutaneous administration of BCG improved BCG treatments. And the concept of priming is basically straightforward. I mean, what priming means is the first encounter of naive lymphocytes, namely T cells, with the antigen. In the human body, this is done by APCs, which means mostly dendritic cells, since those are the professional APCs in our body, which are gonna phagocytose and degrade the mycobacterium and then express in their surface through these MHC molecules a part of the BCG, a BCG antigen. And this is gonna activate and prime the lymphocytes, meaning that they're going to be transformed into a mature state. Now, for tuberculosis, a test is used, which is called Montox, which is based on this concept of the PPD, which is a purified protein derivative. And this purified protein shares some common antigens with the BCG. And there's been studies explained in this paper that point towards a potential of PPD testing as a positive predictor for treatment. They basically explained that PPD screening can be used to predict whether a patient will have a positive or negative response to BCG, though they keep claiming that there is a gap in this research and that further research needs to be done to prove whether this is an efficient way of determining also predictability. Now, moving on to the next section of this paper, which is basically BCG in combination with tumor vaccine. They explored a paper in which a vaccine using this as parts of this protein, which is MHA3, which normally plays a role in the ubiquitination pathway, as we can see here. Though when dysregulated, it can become oncogenic. And because it becomes dysregulated in cancer cells, there is a disproportionately high expression of MHA3 in some types of bladder cancer. That's why in tumor cells, it's going to be, again, expressed in MHC, which can be exploited by our immune system for a better anti-immune response. Now, what they claim is that after vaccination, there's more circulating T cells, right, specific for this antigen. But whether that has a positive outcome for treatment or that can improve treatment is yet to be fully understood. So they call upon more studies regarding the use of such vaccines for BCG treatment. Now they managed to find also some other predictors for response to BCG. So mainly genetic and epigenetic predictors, which are mostly linked to tumor recurrence or treatment failure. And those are going to be ARID1A mutations linked to chromatin modification, epigenetic changes, such as the unmethylation of CDKN2B and MUS81A, estrogen expression, multifocal tumors, which which is to be expected, in my opinion, and a positive fish test. It's not clear to me what precisely they meant with a fish test. I don't know what they are testing for, but they just briefly mentioned that a positive fish test 3 months post-induction was predictive of failure. But just to do a brief mention of the predictors that they found and discussed in the paper. But for me, the most important part, I'm going to explain this later with a diagram I found, is the mechanism of action. So basically, how does biochemically the BCG therapy works, and how can this be exploited to improve outcomes? And there's 5 key points to touch on this area. The first one has to do with the priming, the process I explained before, which is attachment, internalization of BCG by APCs, and this is going to trigger the immune response, right? The immune cascade, which is going to basically help create a better anti-immune environment. And the second one has to do with the recruitment of lymphocytes, more specifically T cells, and the different populations of lymphocytes and their relation with better or worse treatment response. They found that a Th1 dominant environment, a Th1 type immune responses are better for effective BCG, while the opposite, which is a Th2 dominant environment, correlates with failure. And I'm going to show this later in a more graphical way. Then they found that granulocytes, mostly neutrophils, they are able to distinguish cancer cells and attack them through degranulation. And they also found these IL-17 mast cells and some specific populations of natural killer cells or NK cells have a higher expression of CD56 and CD16, which is going to be the main molecule involved in antigen-dependent cytotoxicity in patients treated with BCG. So this is another pathway which they seek to explore. And lastly is the potential of combination of BCG with cancer vaccines. That's another yet not fully understood area of how BCG as a whole works. And this is going to be very clearly explained in this diagram I found, which tries to basically show all the different immune pathways involved in the immune response post BCG. And as we can see, so this little yellow ovals is how BCG is depicted. They're going to be phagocytosed and detected by macrophages and dendritic cells, and then based on the cytokine landscape, we're going to have a more Th1 dominant environment or a more Th2 dominant environment. And a key player here is going to be interferon gamma. So this is another possible cytokine involved in the lymphocytic response to BCG, right? And then apart from CD8 T cells and cell cytotoxicity inducing apoptosis and so forth, I want to do a special focus on this type of T cell, which is going to be also very important for the recombinant BCG therapies, which I'm about to mention now. They're mainly focused on the gamma delta T cell, which is going to release a special kind of cytokine, which is the cytokine IL-17. And this is going to be, it's going to promote the infiltration of neutrophils in the tumor microenvironment. And I'm going to show you later why this is of particular importance, since the authors of this paper also explore the role of recombinant BCGs in the treatment of bladder cell and of bladder cancer, and the improvement of clinical outcomes with BCG therapy. So departing from the fact that IL-7A plays a crucial role in the recruitment of neutrophils, which we also know are essential for the anti-tumor response, they evaluated the use of BCG IL-15 recombinant strains which are proven to increase the recruitment of neutrophils and therefore have an increased immune response and therefore also an increased anti-tumor response. So this is one of the recombinant strains that they propose be studied further for a more efficient or more effective response against bladder cancer using BCG, of course. And lastly, this has not been fully dealt with or explored in great detail in the paper, but they do at least briefly mentioned the use of BCG with interferon alpha 2b, which, I mean, although the intracellular pathway or the cytokine signaling to me is not completely clear. It's shown to enhance lymphocyte stimulation and increase cytotoxicity, meaning that it should, in theory, be indicative of a better anti-tumor response. And just to conclude, there's another important role that IL-15 plays, which was not entirely covered in the paper, but I think it's still very much worth mentioning, which is the role of IL-15 in the activation of CD8 and NK cells. And I took this from a paper studying this transcription factor, O2B1. It's not really important for this context, but what's important to know is that myeloid cells, most notably APCs and granulocytes, they use IL-15 to signal CD8 T cells and NK cells to enter an anti-tumor state, right? An increased anti-cancer immunity. So this is yet another way in which these cellular pathways could be used to expand on BCG potential. So, well, that was it. I remain open to any questions you may have, and thank you for your attention.